Hi there. We're two Mareks from Artisan, and together we're hoping you'll have about 15 minutes or so to spend with us on this video to listen about our gene regulation platform. Two words about who we are, and we'll jump onto the matter after one slide introducing Artisan. So I'm the guy on the left, and I have joined Artisan over 3 years ago after returning from the United States, where I completed my postdoctoral fellowship program at Oak Ridge National Lab. I currently work together with a very talented team of bioinformaticians at Artisan on problems spanning single-cell RNA-seq, multi-omics data integration, or biomarker identification, just to name a few. As I was always keen in exploring gene and genome engineering, I teamed up with Marek on a subject that you will hear more about today, the gene regulation platform. Marek?
So I'm the guy on the right. I've joined Artisan a year and a half ago. I did my postdoc at the University of California, Berkeley, focusing on genomics and application of machine learning in translational regulation. It has been pretty exciting for me to work on some of the modules of the gene regulation platform, and I'll be very happy to talk about them today. Thanks.
Thanks, Marek. But first, before we delve into the science, let me very briefly, on one slide really, introduce Artisan to you. So for those of you who haven't come across our company, we were established 5 years ago. So far, we have completed over 180 projects with multiple customers. Naturally, none of those will be possible without our fantastic team of over 110 colleagues. We also operate internationally. Now, onto the subject. Have a look at the slide. Designing efficacious crucible guides, selecting the most efficient SRNA, diving into the complete genetic profile of your target gene. Those are the kinds of questions we find reappearing during our talks with many of our collaborators. They're mostly revolving around the preclinical phase, but also have potential to apply beyond that. There are of course many more, and our experience is not limited to those you can see, but for the time's sake, maybe let's focus on some of those most common. So, were you ever wondering how would it be to have a platform that can answer all of those questions without the hassle of having to work across multiple unconnected tools? The gene regulation platform. A platform that accelerates drug discovery from target identification to the preclinical stage. If you answered yes to any of the questions listed on the previous slide, then listen on. Our platform is here to help you in your day-to-day work. We have distilled our extensive experience into this multi-module platform that provides complete 360-degree overview of gene regulation. It provides 4 modules, focusing on siRNAs, CRISPR-Cas9 probe design, microRNA analysis, and thorough characterization of the genetic landscape of your gene of interest. Today we will show you only 2 out of 4 components of the gene regulation platform, the siRNA and the genetic reports. Marek, could you walk us through the siRNA module?
Certainly. So the aim of this module is to provide access to a global repository of high-quality siRNA molecules for every transcript across multiple animal genomes. Our repository, along with the accompanying app, has been designed with modern and easy-to-use interface. For the purpose of automation and being used as a part of a bigger system, it allows programmatic access using REST-compliant API. In case of the need for updating the system, the database creation process is fully reproducible using Amazon Web Services or local computational server. We have achieved this using the industry standard technologies for pipeline creation and containerization like Docker and Nextflow. Among key challenges this project has faced was the vast amount of data stored in the database. Yet thanks to using PostgreSQL as our database solution, we have achieved near instantaneous app responsivity. The highlight of this repository, however, is the richness of the annotation that was generated for all of the siRNAs. One of the most useful is the ability to check for the cross-species activity of particular siRNA. This allows finding siRNAs that will work both in human and in mammalian cell lines. Other annotations included are off-target activity, effectiveness against splicing isoforms of the particular gene, single nucleotide polymorphisms overlapping the target site. Altogether, this information provides insight into various aspects of successful siRNA design. But most importantly, we have validated our prediction results against a test panel of 2,000 siRNAs, achieving prediction performance better or equal to the leading algorithms in the field. Altogether, the siRNA database provides an easy-to-use, high-quality, and extensible resource, since thanks to the reproducible pipeline, we are ready to extend it to additional datasets or add required annotations as needed. For me, the picture is worth 1,000 words, so please have a look at the snapshots of our user interface, and you'll be able to see it in action next. All right, so you can see the pre-recorded demo. It is showing the help, and you can see and check the topics available for quick reference. You can select the gene and then select the primary transcript for the predictions, adjust the parameters for the predictions, select the length of the siRNA, and press search to see the siRNAs, which will show up after a while. There are also links at the bottom to check out for the particular gene. So you can see the table of the results now. The most important column is the efficacy score, telling how well the siRNA will work. But there are other ones like the shared transcripts, which tell which isoforms of the gene are affected by this siRNA. There are also off-target columns, which describe the off-target genes affected by the siRNA, and additional columns which provide extra information. That's pretty much it. It's perhaps now we can go to your module, Marek.
Thanks, Marek. Let me go to the next slide then. The genetic report can be thought of as an extensive dossier on gene function and consequences of its native variability, such as SNPs, alleles, pathogenic mutations, epigenetics, and other modifications. It's truly comprehensive, encompassing over 2,000 of data sources. The best feature is that while it summarizes findings into easily digestible written narrative, it also enables to export all the underlying data through the use of enhanced tables. There's a whole battery of plots, and the more complex ones are also interactive. You can zoom in when it's necessary to get a better overview of the data. The rich living document is our most popular product, being a great start for the investigation of a prospective drug target candidate. The genetic report is built of many chapters, starting with the most up-to-date literature overview using most credible scientific sources, going through the variance characteristics and looking at its associated traits. If it sounds like a little bit of too much and you need to catch a breather, we have a summary and executive summary for you too. Here you can see two snapshots of the genetic report interface. I'm going to walk you through in a similar way Marek did just a second ago through a demonstration version as I'm browsing through the chapters. So here's me going through some of the chapters, gene and protein overview to start with. You can see tables appearing, various kinds. There are many chapters, and you can click on the figure, it will pop up, providing better view. Associated traits is one of another chapters. All those tables are searchable, they're sortable. The graphics, you can really interact with them. You can click on dots, you can click on the legend. You can have a look at the gene expression patterns across various repositories. And as I mentioned, the graphs are truly interactive. You can select a fragment of it. You will get a zoom-in version. You can hover over your mouse over a dot to get a better overview of some of the p-values, perhaps. Many different kinds of plots, really. And perhaps you'll also be interested in downloading and exporting some of the data sets here, some of the data to your favorite file format. Okay. So the gene regulation platform, a platform that accelerates drug discovery from target identification to the preclinical stage. If you found any of the presented modules today interesting, or you'd like to hear more about miRNA module or CRISPR-Cas9 sgRNA probe design, we'll be more than happy to connect to you to talk more about it. Here you can see the contact details. Okay, thanks.
Thank you, bye.